everyone, and welcome to our video blog. We are 4 PhD students, and we are part of a ITN called ViBrANT, and we are studying how virus and bacteria adhere to the host cell. Now we are going to talk about how do we study proteins. And for this, my name is Diana, and here I have Andreas, Sounak, and Anchal. We would like to start by talking about what are proteins. The human body contains about 40 trillion cells, and each cell is composed of 40 million proteins. Proteins are molecules which are composed of amino acids, and these amino acids are connected to each other, forming peptides. These peptides arrange in a very complex structure, which is called protein. Now, proteins accomplish very important functions in the human body, and they are also present in other organisms like bacteria and virus. Studying them, it's important to understand disease, but also to find new therapeutic approaches which can help us to target these diseases. A nice example we had recently with the description of the spike protein, which is present in SARS-CoV-2. After the study of this protein, we could develop vaccines, and we can also develop therapeutic approaches. But proteins are too small to be studied by the simple eye. So we have very sophisticated techniques which are used to study them. And for this, the experts here will drive us to the different techniques and explain us how do they work. I would like to leave the floor to Andreas so he can explain us a bit more about X-ray crystallography.
Okay. X-ray crystallography has been around for quite a while, and it's a fairly robust technique to figure out how proteins look like on an atomic level. And you can imagine proteins as like molecular machines. So every individual part of the protein is important, how it is arranged. And how to figure out how they look like, we use X-ray crystallography. So in that, you first need a protein crystal, which is a very ordered arrangement of proteins. And then you use a complicated mechanism, which is X-ray diffraction, whereas you record all the spots on a detector, and then via computational methods, you get to what is called the electron density map. And then you actually have to build the model so that fits with the data. And this method is really important because it can tell us exactly interactions between proteins, for example. In the example that I put here, that shows the SARS-CoV-2 binding to a human receptor. And you can see exactly what kind of interactions are important, and that you can, for example, develop vaccines and you can develop drugs. So that's why this technique is important to us.
Thank you, Andreas. Now we will leave the floor to Anchal, who is going to talk about CryoEM.
Thank you, Diana, for the introduction. So cryo-EM is another method to deduce the atomic structure of the biomolecules, especially big protein complexes that are hard to crystallize. Here we spray purified protein samples onto the mesh grid, followed by blotting to remove excess of the sample. And this will then need to be flash frozen in liquid ethane, also known as cryogen, for cryo-EM. The grid is then put in the microscope and subjected to the electron beam. The electron detector generates single particle images of the molecules embedded in the thin ice in different orientations. The image processing software apply motion and contrast transfer function corrections, and the individual particles are then selected and aligned to calculate 2D class average. This will then use to generate 3D classification, generating the best 3D map, and finally calculate the high resolution cryo-EM structure. Thank you so much.
Thank you, Anchal. Now we want to hear about mass spectrometry, and for this we have Sounak, who will give us a bit of information about what is that.
Thanks, Diana, for the introduction. Yeah, so as we heard from Diana, that proteins are quite an integral part of all living individuals or living organisms. And as you can see from the left side, I have tried to put down some of the points where proteins are quite important in all biological process, like immune regulation and cell signaling, and they also play an important role in catalyzing important reactions. So therefore, there has to be a method as to how, or an instrument as to how we study proteins. And for that, we use a machine which is known as the mass spectrometer, where you can see in the figure, this is a mass spectrometer. And why do we use this mass spectrometer? We use this mass spectrometer for identifying proteins in a sample. We try to look how does this proteins look and what are the different modifications within this proteins. And also we try to look what is the sequence, as Diana said, that these proteins are made up of different amino acids. So what are the different amino acids within these proteins that make up these huge proteins? And how do we do that? So we have a method, and you can see from the schematics below, if you see there is a particular protein, and then we start with, this is one of the basic methods that we do on a mass spectrometer. So we have a protein, and then we come up along with an enzyme that's more like a trypsin, which will chop the proteins into peptides, like Diana pointed out, and these peptides are then put into a mass spectrometer. And then different software or bioinformatic tools are used to identify these proteins, or what are the proteins. And a most common example of my method being used here is on the example that I have is, if you look here, that one of the bacterium known as the Streptococcus pyogenes, which infects human beings, and they cause both localized and systemic infection. So if you look on the left-hand panel, when they cause localized infection, they interact with different kind of proteins in the saliva, like immunoglobulins, like IgAs. And on the right-hand side, when they enter the blood, they interact with different kind of proteins from the blood. And the most common is complement protein. And this is what a mass spectrometer helps us to do, is to identify protein interactions.
Thank you very much to all of you for your nice contributions. And now I would like to address some questions. So it seems to me that one single technique is not enough to describe a whole process. And it seems to me that the combination of different techniques, it's the key for further discovery. So I would like to hear your opinion about it.
Yeah, I absolutely agree that the integrative technique is what will help us identify all. I mean, if you want to address a biological question, I think integrative approach is the best way, where you start using mass spectrometer from identifying what kind of proteins are present in my sample of interest. But the limitation is then you actually don't have an atomic resolution as to how this protein interacts with each other. And then you come up with Xray crystallography or cryo-EM, which will actually tell you how these two proteins are interacting with each other. And then these are the ways where you know the protein interfaces, and then you come up with therapeutic approaches. The most common application is, like, if you consider the development of a drug. So the first thing that you need to know is which are the proteins you want to target. So, and then you understand, or you identify those proteins using a mass spec, and then you want to know where actually these proteins interact, and you can do it by using structural techniques like cross-linking mass spectrometry, which is a new advancement nowadays. People have been studying this recently. You can use cryo-EM, you can use Xray crystallography, and then this will actually point out the structural interface, and then this gives you ways for designing new therapeutics or drugs.
And what about when we have a really complex protein which cannot be crystallized? And then how do you describe the structure? This question goes for Andreas or Anchal, if you have any comment on that.
Right, I would like to comment on this. So there are like not only the complexes, but there are some big proteins like my protein that I'm working with, HSF. It's very big and it cannot be crystallized. So with those kind of protein and bigger complex, we employ cryo-EM for that, where we make the sample grids and then we flash the electrons onto it to see the complex in the flash frozen form. So that's how we, and then we get like images for it, and we create a 3D class image, and then we describe the structure for the big complexes in that case.
Thank you. And finally, now we are hearing a lot about artificial intelligence and how bioinformatic tools are changing our way of understanding how science works. So do you have any comment about, Andreas, do you have any comment about any new tools that are available which allow us to jump a bit ahead from hands-on on the lab and just prediction?
Yes, so I think a lot of people heard about Google's AlphaFold recently, and it's very interesting because that it hasn't made it into mainstream media because it's quite a big thing in the field. And for ages, the holy grail in structural biology has always been how can you get from a sequence to a structure without having to employ the classical methods. And this the method AlphaFold comes very close to that. And the benefit of that is that even like layman people can use the tool, which before you had like to have a lot of specialized departments if you're a company and put a lot of money in, because these methods take a long time and it's very unclear if there's actually a result at the end of it. So I think AlphaFold creates this new avenue for companies to go down to, or even for like academics to just use the tool as you would use any other tool without having to be an expert in the field. So I think that's the biggest benefit of the technique.
And it seems to me that it's also saving a lot of time, right? Because if you can predict the protein structure based on what is already available, then you don't need to spend all the time in the lab trying to really get it. And yeah, a bright future for a protein study. So with this, I would like to thank you all for the really nice discussion about how do we study protein. And yeah, till the next time.